screen just fine. And you'll be able to see the recording. So let's go ahead and get started. So what I'm going to do, I'm going to talk in two different patterns here. So I'm going to talk in sort of an in-person pattern and then also an online pattern. And so basically when you cycle, yeah, you guys want to jump in, that's fine. Yeah. So we're talking about the cell cycle piece of it. I'll see you guys pop in here. So we kind of looked before at kind of the whole little nebby thing anyway here. So we kind of looked at basically the sort of the whole cellular cycle, right? The G1, S, G2 of the cell cycle, right? And that was interphase. And then we had mitosis and the cytokinesis. Now we talked a little bit about G1, S, and G2, but what we want to do now is we want to kind of start talking a little bit about in particular, the mitosis part of it, right? So the division of the nucleus, and then we'll talk a little bit about control and regulation at the end of the chapter. So just to kind of refresh your memory, G1, S, and G2 is So G1, S, and G2 is interphase, right? Are you on the loop? I haven't. Oh, wait a minute. If there's an admit here, there it is. Sorry. It usually pops up. There it is. So you guys should get it. You good now? Okay. So you don't have to look at my shoulder. Okay. So okay, somebody's mic is up. There you go. Thanks, Marina. Knew it had to be one of you two. Okay, right. So when you take a look at interphase, basically your G1, S, and G2. We talked about that one last time, right? We talked about how G1 was basically like your cellular childhood. This is where you're growing, you're kind of becoming mature. And then once you commit to the cell cycle, then you go into S phase, which is DNA replication. And remember, our mantra is duplicate and then separate. And then G2 is like your preparation phase, right? This is basically when you're getting ready to do your nuclear division. And so you don't want to spend a whole lot of time doing that. So you're doing all sorts of things, right? So you're condensing your chromosomes down. We talked a little bit about that. You're building the molecules that you're needing to do this job, right? So for instance, you need tubulin synthesis. Why? Because your microtubules are what's the tracks that you're going to be using to move your stuff around, right? In this case, your chromosomes. So you need tubulin, you need microtubules to do this with. And of course, you need your organizing centers, right? Your centrioles to do that as well. But there's also another player in all of this, right? We talked about motor proteins. You also need the motor proteins in order to actually move stuff back and forth as well. So all these proteins you're busy making in G2. You want to make sure you have enough of them so that you can get the job done. You don't, the last thing you want to do is to get stuck somewhere in between mitosis and unable to actually finish and complete the job. So that's basically what's all of that, what's happening in interphase. And then of course, in addition to this, right, you're preparing your actual chromosome, right, by adding that little protein pad on your chromosome, the kinetochore, that's right around that little constriction point. That gives you something to grab onto. And so you're kind of organizing and kind of condensing down your chromosomes as well. But basically this is all happening, all these preparations are happening in interphase itself. So ultimately this is what you should look like. So you should have basically your two sisters here. And those are identical copies, right? So this is like mom one and mom one. So this is identical copies of the two moms. They're going to be held together to centromere by cohesin. And then that is going to be where you'll find the kinetochore as well. Right? And then the kinetochore is that little handle, if you will, that your microtubules will be able to grab onto. And so once you've done all that, then you're ready to go. Right? So all you have to do now is just basically move forward. So let's go ahead and take a look at the sort of the stage-wise breakdown of what this looks like. So first of all, we're starting off with the interface. And we've seen these, right? We saw these when we did lab. So these kind of look familiar to you. But here you can see an interface cell. This is a G2. How do you know it's G2? Well, first of all, it's a fairly mature size, but you can also see these nucleoli in here. That basically means you're busy doing protein synthesis. What proteins are you making? Well, you're making tubulin, you're making the motor proteins, right? You're making all the proteins necessary for all the structures necessary to be able to do your cellular division. And so that's one of the things happening. In a G2, a couple of major things you want to kind of keep track of that are happening is, first of all, chromosome condensation begins, right? So you're beginning that process of chromosome condensation. And then the other big thing that's happening, well, we already know that the organelles are dividing, but basically your spindle holes are duplicated. So here you can see your two centrioles, right? They've duplicated and they're starting to branch out, starting to create these long microtubule networks that are going to be necessary for you to be able to move your DNA around. Okay, so once we do that, that's our preparation phase. Let's take a look at mitosis, right? The phase of mitosis. So when you look at the phase of mitosis, there's 4 or 5. Depends on where you come from. Basically, you have prophase, metaphase, anaphase, and telophase. And then, of course, there's a prometaphase. This sort of is a transition between prophase and metaphase. And typically speaking, when you take a look at the presence or absence of prometaphase, there's not a wrong way to do it. So like somebody who doesn't talk about prometaphase isn't wrong. And somebody who tosses prometaphase in there isn't right? It's all the same thing. The cell cycle continuum is what it is. These are just artificial lines that we draw on the process to help us sort of define something. But all the same things are happening, regardless of whether we actually mention prometaphase as a separate phase or not. Okay. Now, the reason why I like to mention this is because depending on who you are, what your background is, you're going to either use the 4 or the 5 system, right? So for instance, if you talk about prometaphase as phase 2, that usually means that you have learned your mitosis from a biochemist or from a cell biologist, because the events that are happening in prometaphase is all that spindle organization, right? So all those microtubules that are growing out, and there's a lot of microtubule dynamics going on in prometaphase. And so a lot of times those microtubule biochemists will really be interested in this particular area of cell cycle. And so they kind of focus on those microtubule development. And so that's why they give it its own phase, prometaphase. But people like me, for instance, have a different background, right? We don't have a biochemistry background, we have a meiosis background. And so somebody who looks at the cell cycle from a meiosis point of view only sees 4. They see prophase, metaphase, anaphase, telophase. But what they do, because they're not focused on the biochemistry of microtubule dynamics, that's not what their thing is. What they're focused on is on the chromosomes and the nature of the chromosomes and how those chromosomes are being separated and sorted out for the purpose of passing on to the next generation. So a meiosis person will only see 4 phases. And then what they'll see is a prophase that is subdivided into subphases. So like instead of saying there's a prometaphase, what they'll say is there's prophase, but within prophase, you have subdivisions like zygotine and leptotine and diplotine and pachytine. And there's different little events that are happening in this area. So what they see is an early prophase, a middle prophase, and a late prophase, and events associated with that. So that's the way a meiosis person looks at it. But what we're going to do is we're going to take a look at what each one of these phases and what's happening in each one of these phases. And remember, like I said before, in order to understand meiosis Okay, there we go. Let's see who's back here. Amber's back. Okay, got everybody, it looks like. Okay. So let's see here, make sure my recording's still good. Okay, and do my screen share. I'm actually kind of glad you guys are in the room because I have no clue what this is looking like to the people online. Okay. Good. Back. Excellent. Okay. So in prophase, right, that's our first step. And so we'll get to prophase. There's three main things that are occurring. Number one is chromosome condensation is completing. So where it started in G2, that's finishing up in prophase. So it kind of goes over those lines. And this is kind of where you get that night crawler look, right? So where they're kind of all fat, pudgy, you see lots of space in between them. That's this phase. That's what's going on here. That's because your chromosome condensation is completing. Then not only, but your spindle duplicated in G2, but now what you get is migration, right? So this is now you get spindle pole migration. And the spindle poles are going to migrate into their antipolar position. Right, so you're going into this sort of antipolar position where you have all of your microtubules kind of going out from your two spindle poles, right? That's kind of what you're going for. And then the other third thing that we do is our nuclear envelope breaks down, right? So basically when you break down your nuclear envelope, then you're going to be getting access to your chromosomes so that you can move them around. So the idea is between the three of these, then you should have everything you need to move, right? First of all, you've got your chromosomes in a nice packaged package, right? So like we had the fine China effect, right? So it's packaged into a nice little box. So it's able to move around. In addition to that, at the same time, you've organized your entire spindle. So you got microtubule tracks laid down. You know exactly where you want to move this stuff. And that's all in position. And then you break down the nuclear envelope so you can actually do the attachments and actually pull those chromosomes to either side. So this is kind of what it looks like. So here you can see you have your prophase cell. And you can see that kind of fat, sort of gummy worm looking texture, right? So those kind of fat little nightcrawler looking types of chromosomes with lots of space in between them. And notice you also see the absence basically of a nuclear envelope. So there's no nuclear envelope there. No, it hasn't lined up yet, right? It hasn't finished out. It's just getting started, right? So all these chromosomes are still large and kind of all in a big lump, right? But here you can see your spindle poles are moving into an antipolar position, and your nuclear envelope is starting to break down. Now, before we talk about prometaphase, what I want to do is I want to talk about metaphase first, because I want to talk about where we're going, and then we'll talk about how we get there, okay? So where we're going for prophase is to metaphase. And remember, in metaphase, what's happening is you have your spindle poles, which are really nicely well aligned. And they're all attached to your chromosomes by the kinetochore, and they're aligned up in the middle of the cell along a fictitious line called the metaphase plate. So they're all nice and lined up in single file here. And you can see your two poles are well attached. So that's where you're going, right? So you want this nice alignment of chromosomes down the middle, but that's not where you started. Right? You started with this. You started with that big pile. So how do you actually get from this, just a sort of the random pile, to ultimately that nice line? Well, that's the story of prometaphase. So what's happening in prometaphase is the development of this lineup, essentially. So your spindle poles have already migrated in their antipolar position. And so what they're doing is they're basically sort of extending out their microtubules, and they're looking, fishing, if you will, for kinetochores. And so a lot of times I kind of use like the fly fishing mechanism for this. If you've ever seen anybody fly fish, the spindle poles look like a fly fisherman. So if you ever watch a fly fisherman, they cast out, pull back, cast out, pull back, cast out, pull back, right? So if you're not familiar with the technique of fly fishing, you would think that this poor fisherman doesn't know where they're trying to launch their line. Well, it's not the same thing, right? If a bait fisherman looks for a spot, strikes it, and then lets it sink, right? But a fly fisherman is a little bit different because they don't use bait. They just use the lures that they create a lot of times. And what they do is they cast out, but what they're doing is they're teasing the top of the water. They're kind of touching the top of the water and they're trying to get that fish to get excited about the lure, which they think is a fly. And they're trying to get that fish to instinctually launch at it and grab it. So what they're trying to do is they're trying to basically use this casting mechanism, this forward, backward mechanism to tease the water to get the fish to grab onto it. Well, the spindle poles look very similar when they do this, because what they're doing is they're building the microtubules as they cast it outward. So the microtubules are basically growing outwards. And in some cases, those microtubules will not find anything, in which case what they do is they retract it back and they cast out in a different direction. And then maybe they don't find anything and they retract it back. And they cast out in another direction and they don't find anything and they have to retract it back. Okay, thanks. Yep. So they're kind of constantly casting out and retracting back. So every now and then, however, when they cast out, then they're going to find a kinetochore. And when they grab it, then they've got a hold of it, right? Just like if you got the fish, you got it on the line, right? And then what they do is this one pole then waits for its other sister pole to grab onto the other side. Now, once they do, what they do is they're kind of pulling against each other like a tug of war. They can feel each other's tension. And what they're doing is they're kind of slowly sliding this chromosome until eventually they kind of line it up right here in the middle. So those are the events of prometaphase. It's a transition, if you will, that microtubule fly fishing dance that gets you ultimately to where you want to go, which is metaphase itself, which is the alignment along that metaphase plate. So next step. Anaphase. Now that you got a hold of them, you got them lined up. Now you have to pull them apart. A couple of things. First of all, you're going to be splitting these sisters apart at the centromere. Well, remember, these sisters are held together by that protein glue, right? Cohesin. So in order to get these things to separate, that cohesin has to let go. So this has to let go. When it does, and you're pulling on the other side, then it'll pull apart. Okay. However, there are two different types of anaphase movements. It's kind of a very simple system, right? So here's your sisters right here. There's your cohesin. So if you have your kinetochore on either side of it, and your microtubules are attached to them, when they pull in this direction, and you let the cohesin go, then this sister is going to move in that direction, and this sister is going to move in that direction, right? So you'll have separated them out. But there's two different movements. Remember we talked about before when you were organizing your microtubules that you had two types of microtubules. You had some microtubules that were attached to the chromosomes, right? And then you had another type of microtubule that I call thoroughfare microtubules that were not connected to the chromosomes, but they were connected to each of the spindle poles, like that. Remember we saw that with the protists? We saw that with the yeast. We even see that with us, right? So there's two different attachments. So and they have two different types of motions to them. So in anaphase A, these are the microtubules attached to the chromosomes. So that's these guys here. So it's a pull. So this gives you like a pull, sort of in a function, right? You're pulling the chromosomes toward the poles. But the thoroughfare chromosomes, these guys are pushing against each other. So these are where the two poles are attached to each other, push away from each other. So this mechanism is a push-off type of a mechanism. So you have two different things. You're pulling with one hand the chromosomes towards you, and with your other hand, you're pushing against your sister pole. You're pushing away from your sister pole. So there's two different motions going on. And this is kind of what it looks like. So here's a classic anaphase. So here you can see the pull, anaphase A is here. These guys are pulling toward your spindle pole. So that's your anaphase A. But the thoroughfares are pushing against each other. So the effect of that is to push the spindle poles away from each other. What that does is this is anaphase B. So it elongates the cell. And prepares it for cytokinesis. Because we already saw the elongation of the cell happening with the bacteria already. So this is an elongation of the cell in us, or in plants in this case. But this is the kind of the dead ringer. So here you can imagine, you can see the spindle poles are attached to each one of these little sides. So you can see they're just pulling on these little chromosomes here. These microtubules are attached to them. So it looks something like that. So the last step, telophase. Now what you do is you basically reverse prophase. So in this case now, you should have one spindle per cell. Maybe you haven't split the cell yet, but one spindle on either side. And so the whole entire apparatus disassembles. So this is basically microtubule breakdown. Where you tore down the nuclear envelope in prophase, you now reform it around your chromosomes. And where you had chromosome condensation in prophase, you now begin to decondense. And as soon as you decondense, guess what you do? You get to work. That's your nucleoli start to appear. This is a sign of a cell getting back to work. Right, because it's making proteins. And that's what it does for a living. Here's what it looks like. You can see basically you have two dense clusters of nuclei between each other, typically still separated by these thoroughfare microtubules in between. And remember what we said before. Remember we talked about the bacterial septum, where we talked about FITC was the protein that sits those two cells apart. Remember we said that FITC resembled a particular protein in animals and plants? That protein was what? Tubulin. And what do we find here in the middle after telophase and cell division? We find a bunch of microtubules made of what? Tubulin, which is FITC-like. So basically, you have essentially the components of a septum there. It's already there. Then, of course, you can see the cell starts to elongate, and then now you just have to enter into cytokinesis. Okay. So once you enter into cytokinesis, then basically we see two different systems, right? One that we see in animals and one that we see in plants. In animals, the addition of actin filaments in that little septum area. And that forms what we call the cleavage furrow. And in plant cells, we see the cell plate, right? We talked a little bit about this in lab. So here you can see the cleavage furrow happening in animal cells. So basically you've got the addition of actin in addition to the microtubules that are already there, right? So the microtubules are already there from the thoroughfare microtubules. And then you add these actin rings as well. That creates another little contractile ring that also pinches the cell apart, just like we saw with bacteria, essentially. And that'll pinch the two cells apart into two individual cells. Now, this is kind of sometimes it helps to see what an actual cleavage furrow looks like. These are actually real cleavage furrows. So here's a cell, you can see this little pucker right there. That's a cleavage furrow. So you can see how it's basically getting cinched apart. It's going to get pinned apart. It's almost like you have a little cord right in there, and you're just like tightening the cord, and you're pinching it apart. This one here is a very, very deep cleavage furrow. You can see it's nearly completed. It doesn't have very far to go before it gets pinched apart. So this sometimes these cleavage furrows, when they get to this stage, they can look like a lot like a taffy pull, like you're just kind of pulling a piece of saltwater taffy apart. Kind of got that nice little thin isthmus in there in the middle. And then, of course, this may refresh your memory on when we talked about plants, right? So in the case of plants, instead of using the cleavage furrow, you use the cell plate. So you take Golgi-derived vesicles with plant cell wall material in them, and you line them up in the center, and then you fuse them together. So these all start to fuse together to create your cell plate. And eventually this will kind of fuse together into one long cell plate, and it'll look something like this. And then once you fuse that cell plate together, you just fuse it to the external wall, and you've got your two individual cells. So different mechanism, but they still kind of get the job done. And yes, I do apologize for being a mess. Still, it's funny how quickly congestion sets in and how long it takes for it to clear out. What? Yeah, well, that's because it's all like, it's right here, it like landed right on my vocal cords, and it just does not want to leave. So I sound terrible. I don't really feel that bad, but I sound terrible. So guys, like, just stay away from me. Like, okay. So now that we have the cell cycle in mind, now remember, this is a cell cycle, honestly, that you want to know well, right? So remember, like we said with meiosis, if you want to understand meiosis, you want to understand mitosis well. And the way you do that is by understanding what happens in prophase, what happens in metaphase, what happens in anaphase and telophase and cytokinesis. So the more you can rattle that off and say, like, these are the events at each one of these steps, this is what's happening in each one of these phases, the more comfortable you can feel with that, the easier meiosis is going to be. Because when we get to the next chapter of meiosis, the events are largely the same. The difference is that there's just a couple of additional tweaks that you throw into meiosis that makes all the difference. Okay. So really, studying the cell cycle, if you do it right, is like studying 2 chapters for the price of 1. And that ain't bad economics. Okay. So now that we actually have cell division, so we understand interphase, we understand G1, S, G2, we understand mitosis with prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis, and then back to G1 again. So now that we understand that, we need to understand how to control it, right? Because if you think about it, not every cell in your body is divided, nor should it, right? But some cells are. So like, for instance, your skin cells are actively dividing because there are a lot of wear and tear there. You do a lot of damage to your skin cells, you constantly have to replace it. So that makes sense that those are under heavy mitosis. If you injure yourself, it'd be nice to have mitosis around so that you can replace those damaged cells. So the one thing, though, that we need to really be crystal clear on is that cell division is a dangerous thing. It can be, right? Because it's not okay to just divide. I mean, it's not like, this is kind of where cell division, I mean, our analogy of us making a decision to have kids is like cell division kind of a thing. This is kind of where that analogy breaks down. Because technically speaking, for the most part, if somebody wants to have 25 kids, they can. Now, I'd question their sanity, but there's no harm, no foul against that, right? I mean, there's no problem with that. If you want to have a little family or a big family, there's no consequence to that other than maybe if you have 25 kids, your kids are probably raising your kids and not you. But anyway, and don't ask me how you're feeding them because I'm having a hard time with 3. I can't imagine 25. I mean, I guess you just send them out to pasture and just tell them to find something to eat and hope they don't die out there. But anyway, that's just me. So the idea, obviously, then with the cell cycle is there is a consequence, right? So it's a little more controlled than that. So you can't just divide and have as many cells as you want. You only divide and have the cells that you need. So cell division is only done when you're told to do it. So in a lot of ways, that restriction point isn't really like us, is it? Because we're the ones who make the decision as to whether or not we want to have kids. But a cell that's in restriction phase doesn't make that decision. That decision is made for it. So there are molecules that basically will tell that cell whether or not it divides. This is kind of like, for us, this would be like a really strong totalitarian state, right? Where the government literally tells you, you can have 2 kids, and that's it, right? I think China's getting close to that, right? But guess what? You didn't make the decision, did you? The government made it for you. And not only that, but if you make the decision against the government, good luck. I wish you luck, because you'll end up basically doing hard labor in some godforsaken outpost somewhere. The Russians were great at that, right? They love that Siberian gulag system. It's like you're going out there, you'll either work yourself to death or you'll freeze to death. But either way, you're going to die. That's not an option, right? It's just a matter of how much do you benefit the Russians. So that's kind of a little bit different, right? So that kind of gives you a sense of sort of the control of cell cycle. So cells that are dividing in your body aren't deciding for themselves. They're having that decided for them. And so there's a lot of control there, right? And there's a reason why. Because the cell cycle can be quite damaging if you don't do it right. And none of that, but it's wrapped around 2 unforgivable points of no return. And these make sense, right? So once you replicate your genetic material, guess what? You're in it to win it. You don't have a choice. You can't walk around with twice as much DNA. You got to deal with that, right? So that's a point of no return. And also, once you separate your sisters, you're also in it to win it. As you're going through anaphase and you're separating your sisters, there's no oopsies. There's no, wait a minute, can I redo that one? No, sorry. You have what you have. You are what you are. You live with what you are. Good, bad, and indifferent, right? The problem is, if you didn't do that separation just right, then you're living with a whole lot of bad. And if you're a multicellular organism, that's not a good thing, right? So because you have these unforgivable points of no return, then what we have is essentially a system that has a lot of heavy regulation to it. And your cell cycle is controlled in the progress of the cell cycle at specific points called checkpoints. And so at these checkpoints, they're kind of like they're like a border crossing, where you kind of have these checkpoints where you stop, they check you down, they check to see, who you are, what are you doing here, that sort of thing. But basically, these are cessations, pauses, arrests in the cell cycle. And basically, they do certain things. A lot of them are going to be checking for accuracy of the cell cycle, make sure you're doing everything right, and to see if you're picking up any errors. Now, we kind of alluded to that before, right? Because we said S phase, one of the things that happens in G2 is that after you go through S phase, you're doing a lot of DNA repair, right? So there's a lot of repair in there. There's a lot of errors that are made. But ultimately, this allows you to sort of pause and sort of clean some of that mistakes up. That way you can kind of move forward. That increases your accuracy. However, another thing that these checkpoints will do is allow the cell to listen to instruction. Both internal instructions and external instructions. So internal instructions, like if you were for us, would be like, am I ready to have kids yet or not? That's like an internal instruction. Then you tell yourself, yeah, I think I am. I think I'm ready to do this. I think I'm ready to have kids. That's like an internal instruction. That makes up your mind for yourself, whether or not you have the body and the maturity to pull this off, okay? An example of an external instruction would be like the Chinese government telling you that you can only have one kid. That would be like an external instruction. Now, I'm just picking on the Chinese because I know they've got policies like that over there, but that's kind of like a combination of the two. So it allows you to be able to listen to what you're supposed to do. And the important thing is that in the cell cycle, there is a very strong mantra in the cell cycle. Do what you are told. That's it. Guess what? If you're not told to do it, what do? You don't do it, right? As a matter of fact, you just basically sit there like a bump on a line until your instructor comes in and tells you what to do, okay? So how are these checkpoints, these regulation points organized? Well, you have 3 of them. The first one we've already talked about, actually. That's the restriction point. The G1 it was actually referred to as the G1S checkpoint. This is where the decision is made for the cell as to whether or not it divides, okay? Now, in addition to this, telling the cell whether or not it should divide is a bunch of external signals because the cell itself doesn't really know if it's necessary to divide or not. It can determine whether or not it's capable of dividing, that's true, but it can't tell whether or not the tissue needs an extra cell or not, right? That's why the external signals come in and say, you know what? We're a little shy on tissue out here. We need replacements. You need to divide because we need extra tissues. Right? And then there was one. We're dropping like flies here, which is not difficult because there's only 2 of us to start with. Right? So the next one is what's called the G2M checkpoint. So this basically is in G2, right on the precipice of starting mitosis. So this is where you're preparing for mitosis. So all those events that we talked about in G2, like building your centriole, duplicating your centriole, building your tubulin, building your organelles, doing your DNA repair, that's getting ready for going through mitosis. That's what's happening at this particular checkpoint. You're stopping the cell cycle, you're pausing it to give yourself time to make sure that you got everything you need before you go into mitosis. And so this is where you can do a lot of that DNA repair. Then once all that looks good, you move forward. You go through prophase, through metaphase, and then you arrest at metaphase. This is called the spindle checkpoint. What you're doing here is you're testing for good connections, right? You want your microtubules to be attached to the kinetochores and for good balanced tension. So remember, your spindle is attached to the sisters. So this attachment has to both be a good sound, solid attachment, but it also has to have equal tension on either side, right? So you're kind of testing for a balance of tensions. That's a critical thing for separation. Because if you have an unbalance of tensions, if there's one side that's stronger than the other, then that strong side is going to get everything. And the other side is going to get nothing, right? So if this side here is stronger, what's going to happen is both sisters are going to move to this side and nothing's going to be on this side. When you get that unbalanced separation, which is due to unbalanced tension, that unbalanced separation is called non-disjunction. And notice what it does. It gives one cell too many chromosomes and the other cell not enough. Remember we said a few slides back, we said that for animals, in particular us, we don't tolerate extra or missing chromosomes. So this would be an example of an intolerable outcome. So what would happen to cells like this? They would die, right? And you would have to start over again. So you arrest it at spindle formation, at metaphase, just to make sure that everything is attached. It's kind of like if you're a truck driver and you're tying down your load, right? Right before you jump in the truck to get ready to roll down the freeway, you're kind of pulling on all the straps just to make sure they're all good and tight. You're checking your load to make sure there's good balance of tension, that nothing's going to slip off your pallet, right? While you're driving. So that's kind of what's happening here. You're sort of pulling on these microtubules, checking the load, checking the attachments, making sure the tensions are balanced before you separate. Because remember, we said that once you separate, that is a point of no return, right? There's no going back from that. So this is going to be an example of like, if you do this unbalanced separation where everything goes to one side, nothing goes to the other, sorry, you're stuck with it. You're living with it, right? Because that's a point of no return. You can't undo what you just did. You can't unseparate the separation. There's no mechanism for that. It's kind of like you can't untoast the toast. Once it's toasted, you've got toast. The only thing you can do is throw the toast away and start over, right? So that's basically how these checkpoints work. So here you can see here's your G1S checkpoint, is your restriction point. Your G2M, which is right at the doorstep, getting ready to start mitosis, and your spindle checkpoint, which is right in the middle of mitosis itself. So one of the things I always like to do for students to kind of give them an analogy of how this sort of control works is I always like to use an analogy of like an airport. And I'm going to kind of park it on this screen because this kind of shows you what we're looking at. So has anybody ever actually listened to the communications on an airplane between the cockpit and the air traffic control tower? Right? Because back in the day, I mean, sometimes you can hear that interaction, A lot of times you can't now because of the whole 9/11 thing for security purposes. But before 9/11, I remember when I was in grad school, I was flying back to California for the holidays. And one, they have like the little in-house music station, different channels and things like that. And one of the channels that they had on there was the cockpit channel. And I listened to that, being the nerd that I was. No, I wasn't like a normal person listening to music. I was listening to the cockpit channel. That's what I did. I didn't see those. Sorry, guys, for the late admit. I didn't see it. Doesn't pop up when I'm on screen. So basically what I did is I just listened to that. And the nice thing about it is like that communications is you heard what was happening in the cockpit, not between pilot and co-pilot. But what you heard was the communication between the pilot and the air traffic control tower, which is really fascinating. But you didn't just hear your pilot. You heard all the pilots. So it was like tethered into air traffic control, like open airwaves. So you could actually hear the air traffic control tower checking down flights, like rolling through the flights, like they were all parked at the gates. And of course, you knew your flight number, right? United 50, something like that.
You finally let me in, Biggie?
Yeah. Yes, I did. So basically what happened was as you were sitting at the gate, you kind of heard the air traffic control tower kind of checking down. And they would say like,
I could tell that you've been crying all night, drinking all summer.
Praying for your happiness. I hope that you recover.
Yeah, that's one that needs to be muted. Yeah, that was it. That was entertaining. Yeah, that was helpful, right? So basically what you kind of heard was this sort of check down as air traffic control tower checked on all the different flights and their status, right? So they would be checking on their boarding status initially. You could hear them going like, Continental 1520 status, and then the pilot would check in Continental 1520 boarding, and then the air traffic control tower would say Continental 1520 standby, and then it would start rolling, United, and then they would hear them, and it would say finally it's like, okay, United, close doors, and taxi out to B2 and standby. And then what would happen is you would hear them rolling through each of the different planes, saying the same sort of instructions. And eventually what would happen is the door would close, you would see the gangway pull away, and then eventually you'd see your plane pulling out. And then it would basically taxi over to, and as you looked out the window, because they were like the window seat, you would see down there marker B2. And the plane would sit there. And then you'd hear them talk to all the different planes. You'd say like, American Airlines 510, taxi out to A1 and stand by. And then you would see an American Airlines plane taxi out to A1 and then stop. So you knew that must be 510 over there, right? And so you heard all these different planes happening. And then eventually what would happen is you kind of get in line. It's like, okay, United, whatever my flight number was, taxi into A1 and stand by. And then you hear them rolling through there and be like, okay, American Airlines 510, taxi onto runway, you're clear for takeoff. And then everybody else would be standing by. And the important thing that I got from that, the fascinating thing was, first of all those pilots were well trained. All those pilots knew how to fly planes. They didn't need air traffic control tower to tell them how to taxi or how to move the plane around, or God forbid, how to fly the plane in the first place, right? So what was crystal clear was that every single one of those pilots, even though they're well trained, they could have taken off whenever they wanted. They had the skill set to do that. But nobody on that airport tarmac did anything unless air traffic control told them to do it. So nobody did anything. Why? Because if my pilot said, screw this, I know how to take off, I'm going to just take off, what would happen? Yeah, right? because my plane would take off. They wouldn't be able to see another plane coming in for a landing. We'd collide in midair. Hundreds of people would die. And the pilots wouldn't see any of that. But the air traffic control tower can see all of that. They can see everybody on the ground. They can see everybody who's approaching for landing. They can see everybody who is basically on, multiple decks above the airport who are just coming into the airspace and getting ready to form a circling pattern. Some that are a few decks down that are in a circling pattern, waiting for their turn to approach for landing. And they can see the approachers that are getting ready to touch down. Not only that, but the air traffic control tower can also see the flybys. These are the planes who have no intention of stopping there. They're direct flights from East Coast to West Coast, and they're just flying over. The captains and the airplanes can't see any of that. So if they were to go rogue and fly on their own and not listen to control and regulation, people would die. The cell cycle is very much the same way. Does the cell have the capacity and the ability, the skill set to divide? Yes, right? But what happens if they do without listening to their controllers? People die, right? So just like with airport air traffic control, the cell's highly controlled. So the cells are only listening and doing what they're being told to do, just like every good pilot at an airport, right? Nobody wants to die in a plane crash. Oh, nice. And they crashed?
No.
Yeah. That's why you don't do that. That person's probably not going to be flying a plane again.
Yeah, no, I don't think so.
Well, FedEx is international, but I mean, I can't talk for other international organizations, but yeah, if you're in the US and you take off and you come within a narrow miss because you're not doing what you're told to do, you're not going to fly again. Your feet are never going to leave the ground again. Okay, so let's take a look then. So since this cell cycle is highly controlled and regulated, then how do we go about doing this, right? Remember, there's first of all, one thing you need to keep crystal clear in cell cycle. There's a mantra in cell cycle. Do it right or die. Those are your options. So cell cycle is really not for the faint of heart. It's a zero fail mission, but it's necessary, right? So these are pretty high stakes here. So how do we do this, right? How do we control this? And this is kind of one of the big things we noticed, right? Because one of the things we noticed was that if you're doing cell cycle, and you start ignoring control, right? That means you start dividing whatever you want. You start doing however fast you want. You stop listening to external controls. You don't give enough proper time to fix all your DNA that you screwed up. So a cell that no longer listens to cell cycle control turns into what? You all know it. Some of you know it well. Some of you have family members that have it. Some of you have family members that have died of it. Some of you guys actually may get it for you. Huh? Yeah, no, that's why I wrote it down. Yeah, it's cancer, right? Cancer is basically uncontrolled cell division. That's basically what cancer is. So that's what we say, right? Do it right or die. That's true for the cell, right? Because it's high stakes. Because if you don't do it right, what happens? If you don't do it right, you're going to turn into a cancerous cell, and that's going to kill the organism. So it's better then that if you're a train wreck of a cell and nothing's going right, it's better that you die and that we can build another cell than that you become cancerous and you kill the organism. So with this high stakes in mind, then how do we actually pull this off? Well, there's a couple of things we're looking for, right? We know this is a cycle. We know that we've got G1S, G2MC, G1S, G2MC over and over and over again. So what we wanted to look for was a protein. That's what we're thinking of, right? A regulatory protein. That has a cyclic pattern of expression. So this is what we wanted to see, right? So we wanted to see some sort of a protein that here's our G1S, G2MC, G1S, G2MC, that. And then this is our protein that we're looking at. So we wanted to see, like, if there's a protein that's involved in regulating, let's say G1, then we'd expect that protein to be highly concentrated at G1, and then eventually it'll sort of peter out because it's not needed anymore. And then the next time we see it again, will be highly concentrated at G1, it'll peter out, and then we'll see it at G1 again. That's what we're looking for, right? This kind of cyclic expression of protein concentration. But we also wanted to see that with phosphorylation activity. And here's the reason why. Because what we noticed about this time we were studying these proteins is that the way that regulators regulate other proteins, right? The way that they turn proteins on and off is by a very simple process of adding a phosphate group to them. Okay, so what a kinase basically does is it adds a phosphate to another protein. And this has two effects. It either turns a protein on or turns the protein off. What we noticed was that this was the underpinning mechanism of cell cycle control. The proteins that were involved in cell cycle control were proteins that were turned on by phosphorylation. So what that told us was that the regulator was likely a kinase, a protein that phosphorylates others and turns them on or off by phosphorylating them. So not only did we want to see a cyclic protein, but we wanted to see that cyclic protein also be a kinase. So here's the problem. We did identify a protein that does the kinase activity, that does the phosphorylation. And we called it a CDK, a cyclin-dependent kinase. The problem with that is cyclin-dependent kinases don't have cyclic behavior. This is what a CDK looks like. So a cyclin-dependent kinase is constant concentration, not cyclic. But we did find a protein that did look cyclic. They're called cyclins. So cyclins were cyclic, like this, but they're not kinases by themselves. CDKs were kinases, but not cyclic. So in each case, we had half of the story. We needed both of them to coincide. So we were kind of a little stuck with that one for a bit until we figured out something that was incredible and that kind of helped us sort of move past this. What we realized was that the two of these together actually are necessary. So for instance, cyclin by itself does nothing. No kinase activity. The CDK by itself, no kinase activity. But when you add the cyclin plus the CDK together, then you get activity. So think of it this way. You know, those like little pendants, right, that are something like this. You take one half of it, your friend takes the other, right? If you only have a half of it, it's only half a heart, right? You need both of them together to form the full heart. Same thing's here. You don't get activity with only half of it. You have to add both of them together in order to get your full activity. So this gives your activity, and this gives your expression pattern. So this is what it looks like. So when we studied the cell cycle, at our checkpoints, these cyclins were active. So let's take a look here. So at the G1S checkpoint, as we lead up to it, remember, so CDC basically is equivalent to CDK. So that's just a yeast way to say CDK. So that's the kinase, right? It's always there, but it's inactive at this particular case. Why? Because it doesn't have its partner, which is the cyclin. So it needs its partner in order to be active. What's its partner? G1 cyclin. So what happens is on this side of things, your G1 cyclin concentration is low to non-existent, which is the reason why CDC2 is inactive. Once you cross this threshold of the G1S checkpoint, then you make G1 cyclin, and that turns on the CDC2, because now you have the two halves that have become the whole. And that allows you to clear this checkpoint. Okay. Now you go through S and G2. Again, notice you still have your CDC2 still there. But now what you don't have is your next cyclin. In this case, notice on this side of things, you have, it's a mitotic cyclin now, right? So your mitotic cyclin is very low on this side, so you don't have any active CDC2. After the G1 to G2M checkpoint, you basically create active mitotic cyclin, and then that gives the two halves create the whole, and that gives you active CDC2, allowing you to clear the checkpoint and move into mitosis. Then in the spindle checkpoint, it's a different creature. It's basically what's called the APC. This is what's called the anaphase promoting complex. And so this is a complex of multiple proteins that basically will be responsible for checking the tension and the connections. That's what the anaphase promoting complex does. And when it sees that, then basically it will allow passage and clearage of that by activating itself. So it'll promote actually anaphase by allowing this. And one of the ways that it promotes anaphase when it's active is it will active it'll break up the cohesin that holds the sisters together. Remember we talked about cohesin having to let go in order for anaphase to occur? Well, one of the things that breaks down that cohesin is some of the enzymes in the anaphase promoting complex. So all those like little resolution things that have to happen, the anaphase promoting complex is doing that. So that's how basically you have a lot of your cyclins factoring into all of that. So the CDK cyclin complex is important, right? Because this gives you your active kinase complex, and this active kinase complex we call the mitosis promoting factor, or MPF. This is your active kinase. Not only that, but you can actually control CDK itself, which is a kinase, by phosphorylating. So this is actually true. So a lot of times kinases, whose job it is to phosphorylate another protein and turn it on or off, themselves are actually turned on or off by phosphorylation. We see this actually quite a bit. In this particular case, if we take a look at a CDK, you can control CDK by phosphorylating it. There's two major phosphorylation points. So for instance, if you phosphorylate it at this little green spot here, this will activate the CDK. If you phosphorylate at this little red spot, it'll inactivate the CDK. So even though it's in complex with cyclin and is kinase ready, you can still control. You can still kind of turn it on or off by either phosphorylating at the red zone or at the green zone. And that's true for most proteins. So the MPF is really the kinase that we were looking for the entire time. And this is what controls your activity and all the proteins that are involved in that. So the anaphase promoting complex basically will be checking the tensions. It'll be checking the microtubules that are attached to each other. And then it'll make the enzymes to destroy stuff. Like for instance, separase, which is an enzyme that chops up cohesin, is normally inhibited, is turned off. But at this particular point, what happens is that inhibition is released and allows separase to remove cohesin. So here's an enzyme whose job it is to basically chop up or remove that little protein glue piece that's holding those sisters together. And then once you do that, then you can separate these two sisters apart from each other. So one of the things we noticed is that when you're talking about CDKs, there's not just a CDK. There's like a whole family of them. That basically means there's lots of control, right? But they all have slightly different specialties. Think of CDKs as, if you're into baseball, the baseball season, right, as being like a good bullpen. There's not just one bullpen pitcher. Most teams have like a dozen bullpen pitchers. Do they all pitch the ball? Yeah, they all basically throw the ball. It's the same thing, but they all have slightly different variations to them, right? Like some pitchers are really good with the curveball, and some are like really good against left-handers, and some are just like really good with a hard slider, right? And so they have slightly different nuances to them. Same thing with the CDKs. Animals, unlike yeast and other protists and things like that, are notable because they have a lot of external signals. What does that mean? A lot of control and regulation. So it's much more complicated, right? As a matter of fact, if you've ever actually been in an aircraft traffic control tower, you'll notice it's not just one person up there talking to pilots. How you doing today, Charlie? You all ready to go to LA? No, that's not what it's like. It's, from what I understand, it's probably one of the most stressful jobs actually, that a person can have. But if you look at it, it is like a busy, frenetic hive of activity. I mean, you have people talking to pilots, you have people talking to the people who are talking to pilots, you have managers and brass and directors and all sorts of people that are in communications with all sorts of groups. It is a mind-numbing array of chaos is what it is. And those are those external signals. So the more complicated an airport, the more of those external signals. I mean, if you're some sleepy little airport like Centennial, right? Not to pick on the Centennial Airport, but it's cute. Obviously, they're not going to have as much going on in their air traffic control tower as, say, DIA, right? Because DIA is where everybody's coming. And so they're talking to all sorts of people. They're talking to everybody. And they're coordinating everything. And so it's a lot of external signals out of DIA, not so much at Centennial. Or if you have like a rural airport out in the middle of nowhere, I mean, you may have one person in the air traffic control tower. Yeah, Bob's looking good. Go ahead. You can fly if you want. Right? Say hi to Gina for me. That kind of thing. I have no idea who Gina is. I'm just. So right, that's kind of, so you see a lot of, so the more signals you have and the more complicated body you have, the more need for control and regulation you also have. The more complexity you have, the more you can do. And this is just a recapitulation of the previous slide. So who are the external signals? Well, a lot of those external signals are growth factors. And where do these come from? Pituitary gland. In part, not all of them, but a lot of them do. Right, they're part of the endocrine system. So the example they have here is platelet-derived growth factor, right? So, which I think is a good example. The problem is oftentimes students have no clue what a platelet is. So here's what a platelet is. In your bone marrow, where you have the development of your red blood cells and your white blood cells, part of your white blood cell pantheon is a cell called a megakaryocyte. This is a cell that gets really big and rangy. And there's like little chunks and pieces of its cytoplasm that basically break off into these like little pieces, like little shards, if you will, of the cell. They're like little membrane sacs full of cytoplasm from the cell. These are platelets. And what do? So these guys are responsible for blood clotting. So when you injure yourself, first thing you need to do is stop bleeding. Well, it's easy enough to do if it's an external wound and you can kind of put some pressure on it, but not so easy if it's internal. You can't get to that one, can you? So basically, whenever you have a breach in your cardiovascular system, your platelets will basically, they're sticky. And what they'll do is they'll basically glom onto the wound area and they'll kind of create this sort of coagulated, gooey, sticky plug that will basically stop the extra bleeding. And that's the beginning of wound healing, right? But the first step is that coagulation process. And that's basically caused by platelets initially. Well, the problem is, since you're consuming your platelets all the time, you have to constantly replace them. And so as you kind of, so it's constantly happening, right? And the way you replace it is by basically stimulating cell division, because you're going to lose them, you're going to lose the megakaryocytes, and you got to keep replacing them, because if you don't replace them, then you're not going to be able to coagulate your blood. If you don't coagulate your blood, you're going to bleed to death. Okay. By something ridiculous, like a nosebleed or something like that, some internal thing that nobody ever dies unless you have coagulation problems. And this signal for these cells to divide comes from the growth factor, platelet-derived growth factor. So platelet-derived growth factor is basically what tells cells to divide. That's so it can make new platelets to keep a fresh supply of platelets. Now, as it turns out, the platelet-derived growth factor, which is secreted by your endocrine system, what'll happen is it'll actually have a receptor called a receptor tyrosine kinase, which is a receptor, but it also is itself a kinase. That is to say that the receptor both receives the signal, right? It receives the platelet-derived growth factor signal, but then it also itself, here's your membrane, will act like a kinase, which will phosphorylate some sort of a protein. So basically here, your platelet-derived growth factor has a way of turning on this whole phosphorylation cascade. And that's oftentimes how growth factors work. For instance, other growth factors that we see are things like epidermal growth factor, for instance. EGF is the one that's responsible for your skin cells continuing their growth. Fibroblast growth factor, FGF is another one. That's the one that is stimulated when you cut yourself, you damage your skin. To replace that connective tissue, fibroblast growth factor will stimulate fibroblasts to reproduce and lay down new connective tissue so you can fix your connective tissue in your skin. So pretty much anything that requires cell division has a growth factor telling it and maintaining it to basically divide. And so platelet-derived growth factor is just an example of it. So what happens then if you basically throw up a double middle finger to all of these control? Well, if you have controlled growth and uncontrolled growth, then you become cancerous. What does that mean? That means a cancer cell is typified by its ability to divide without regulation. And because it divides without regulation, it becomes abnormal very quickly. And so this is a bad thing, right? And so ultimately then when we take a look at cancer, there's actually, there's an environmental aspect to it, right? Because we all know full well, if we drink the wrong things or expose our skin to the wrong things, it can create cancer, right? We know this for sure. Some chemicals and radiation, for instance. So we know that certain chemicals can cause cancer. However, there is some genetic component to cancer, and that's kind of what this particular piece is about here. There's really two dominant genes that we have to worry about when it comes to cancer. One is what's called a tumor suppressor gene. Now, in order to explain what a tumor suppressor gene is, I have to kind of explain a little bit of genetics here. So what I'm going to do is I'm going to draw a gene, and I'm going to, first of all, diagram it out as a box. So the box represents the sequence of DNA necessary for us to be able to select the correct order and sequence of amino acids. That's what we call the genetic code, the code for that particular for that particular gene. Now, with every gene, that's your protein piece of it, right? There's what's also called a regulatory region out here. So this is like your coding region. That becomes protein. And then this is your regulatory region. Now, immediately driving the gene is something that we call a promoter. So this drives the activity of the gene. Okay. Now, you either turn the promoter on and you make the protein, or you turn the promoter off. You don't. Now, in this upstream region up here, this regulatory region, we have lots of different types of DNA control mechanisms. One of those is an area where a protein can bind called a tumor suppressor. And what the tumor suppressor's activity to do is to inhibit the production of that gene. So that little flat bar means it's being inhibited. It's like I'm putting on the stop sign on you. So this will basically keep the transcription of this gene from occurring. So it's a way of turning the gene off. This is helpful, especially if this gene is a cell division gene. The way you keep cell division genes that are not supposed to be active off is oftentimes with this kind of suppressor protein who will keep the gene turned off. Now, a proto-oncogene is a different type of a creature. In a proto-oncogene, I'm going to have the same system. So let me just redraw the same system here. So here I've got my cell division gene. Got my promoter. And I've got my tumor suppressor. Now, the way a tumor suppressor gene works is if I incur a mutation because maybe I was too close to radiation or something like that, and that mutation is a tumor suppressor gene mutation, and I take that tumor suppressor gene out so I can no longer make it anymore, then what happens is the elimination or the removal of that repression allows the gene to turn on. Abnormally. So if I lose a tumor suppressor, then that allows the gene that it was trying to keep off to turn on now. And it allows it to turn on when it's not supposed to turn on. So that's abnormal. That could lead to cancer. That would be unregulated cell division. In a proto-oncogene, it's a little bit different, right? So in this particular case, my mutation mutates something inside the gene itself. And now the gene is in it's kind of stuck in the on state. Regardless of its regulation. So even though your tumor suppressor is there, it doesn't matter because it's like circumventing that control signal. I like to think of it this way. Think of it as though you are a toddler in a room full of toddlers and you've got plans to do badness. But the problem is there's an adult in the room. Well, there's really two different ways that you can go about doing what you want to do, right? If you want to do some badness. The first way to do this is the tumor suppressor method. I can do all the badness I want if I kick the adult in the room out. So if the first thing I do is eliminate the supervision, then I'm free to do whatever I want. And that's exactly what happens with the tumor suppressor gene. So you get to do what you want by eliminating the adult in the room, which is the tumor suppressor telling you not to do it. It's like, you know what? I'm getting sick and tired of you telling me not to do this. Maybe I want to go over there and kill my sibling. So I'm going to go ahead and kill you first. And then I'm going to be free to kill as many people as I want. Now, we'd agree that's a pretty bad toddler. But that's kind of what you're talking about, right? So you can do what you want to do if you remove the adult from the room, the tumor suppressor. Now, the alternative to that would be the oncogene situation. This is a situation where it's like, you are so bad that it's like, you know what? Yeah, I see the adult in the room. I'm not impressed. I'm going to do what I want to do anyway. So yeah, I can see that. I can see you're telling me not to kill my sibling, but guess what I'm going to do? I'm going to kill my sibling right in front of you. I'm not listening anymore. So say what you wish. Do what you wish. But I'm going to do what I'm going to do. I'm stuck in the on state and I'm no longer hearing instruction. So an oncogene is bad because it no longer listens to regulation. Even though that regulation is screaming in its ear, stop, 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 right? The tumor suppressor situation is a situation where I don't have any regulation anymore because I killed the adult in the room, right? So now I can just do whatever I want. So that's the way these two genes work. Now, what's an example of a tumor suppressor gene? Well, one of the most important genes that we actually have as a tumor suppressor is what's called P53, a fairly unsexy name for it. P basically just means protein 53. It was 53 kilodaltons. That's what it was. That's how big it is. But P53 is an important G1 checkpoint, and it also has some play in G2 as well. But one of the things that P53 does is that this is the protein that will regulate DNA damage and that will stop cell division so that you can actually do DNA repair. There's three main things that P53 does. So it will stimulate DNA repair. So if you screwed up all your DNA, P53 will stop cell division and give you and stimulate other repair mechanisms so that you can fix your DNA. It also activates other cell division. Cell division genes. And the last thing is apoptosis. So apoptosis is an order to the cell to kill itself. Remember our mantra, do it right or die? Well, let's imagine you cruise on through S phase and you made a horrible amount of mistakes. Well, P53 is going to stop the cell cycle. It's going to halt it. It's going to give you a little bit of time in G2 to fix that mess. And one of two things is going to happen. Either you're going to fix it all up and you're going to be fine, in which case P53 will remove the halt and then you'll be able to move forward. Or what's going to happen is you're going to be having a problem fixing your DNA. It's going to be like waiting and waiting and waiting and waiting and waiting. And eventually P53 is going to be like, you know what? It looks like you can't fix yourself. It looks like you are too damaged to be fixed. So I'm going to make the decision now, instead of giving you coverage for fixing yourself, I'm now going to tell you to stop trying to fix yourself, take the knife to your throat and kill yourself. Why? Because the mantra is do it right or die. You obviously can't do it right. So now you must die. And so that's what P53 does. And what this does, sounds pretty extreme, but what this does is it basically prevents bad cells from moving forward. And it prevents the accumulation of mutations. And you can also tell, so this is like the adult in the room. This is the adult that's saying, listen, put the candy down or you're going to sit in timeout, right? Oh, guess what? If I just take you out, I can have the candy and I want to sit in timeout, which is exactly what cancer cells do. So roughly about 50% of cancer cells show a mutation in P53. What does that mean? They took out the adult in the womb. So now they can do what they want. And everybody knows that kids without supervision are dangerous. If you don't believe so, just read the great book, Lord of the Flies. Right? Where kids go from sweet, well-trained little cadets from a private school to cannibalism. That's where that went to. Do it. So we're at about our witching hour. So what I'm going to do is I'm going to start with this little piece here. It looks kind of scary, but we'll use this to kind of clean up, and I'll kind of cover the last couple of slides, and we'll dig into chapter 11. And actually, I only have 2 slides. I could probably finish these in just about a minute or so because there's not a lot going on here. I'm not going to require you to remember all of this, right? This is just to show you what a cascade looks like. So here's your receptor tyrosine kinase. Here's your growth factor. And this is your kinase tail down here. And so what happens is this kinase phosphorylates a molecule called RAS, which then basically when it's activated will phosphorylate RAF, which will phosphorylate MEK, which will phosphorylate ERK, which will then go into the nucleus and phosphorylate retinoblastoma or RB. So this will go on as a transcription factor and turn on other cell division genes. And E2F, its partner, will also turn on cyclins and things like that for S phase. Okay. The story of this one isn't necessarily to memorize all of this, but the story of this one is to see that you don't just have one layer of communication, you have multiple layers of communication. So you have nested hierarchies, these cascades of communication. Why? Because by the time you get down to here, remember, cell division is so important that you don't just want a single message. You want to double that message. It's like, okay, here's a message. Are you sure? Yes. Are you sure? Yes. Are you really sure? Yes. Are you really, really, really sure? Yes. Right? So you want multiple layers so that by the time you get down here to start turning on cell division genes, you're absolutely certain that this message is crystal clear. Divide. Okay. What this also shows you is that mutations in any one of these layers, MEK, ERK, RAF, will create a cancer-like mutation. So it'll create either an oncogene or a tumor suppressor gene type of a scenario. And so proto-oncogenes typically will create these sort of stuck on states, right, where you can't control them. And so this is a situation where your cascade is stuck in its on state and you can't turn it off. Right? Or the tumor suppressor gene, like P53, for instance, was a tumor suppressor. But RB retinoblastoma is another tumor suppressor. And this is a retinal cancer, is what this one is. And actually, we found this by looking at retinal cancer in babies. Because what happens is we noticed that babies they had a predisposition, which basically means that one of the 2 alleles are mutated. So you have a bad RB. You only have one good one. Well, the problem is, since your retina is being bombarded by ultraviolet light radiation, it's very quick for the second one to mutate. And then this baby now has no functional retinoblastoma, which is what causes the cancer. And it has retinal cancer, and these babies go blind very quickly. And that's kind of the story, right? The idea with tumor suppressors is that, typically speaking, is genetically they're what's called recessive, which means you have to have 2 of them in order to get the cancer. The reason why we don't get cancer until we're much older, usually, is because it takes a lifetime to accumulate those 2 mutations. So we'll pick up the first mutation in, say, a tumor suppressor by the time we're 20. It may take another 40, 50 years before we pick up the second one. And then in that case, it just begins the destruction of the tumor suppressor. It doesn't mean you get cancer right away.
It could take years after that for cancer to actually accumulate and develop. Because cells don't just turn on or off. They don't just become evil right away. It's a slow progression, but it starts with the loss of a tumor suppressor or by the gain of an oncogene. So that's the end of 10. We'll start 11 next time.